Hello everyone, welcome back to my channel Competitive Botany. In this video I am going to share APSET Life Sciences question paper 2024. The test was conducted in the month of April 2024. I am going to share the question paper with answers. Okay, so let's see the questions one by one. In the last session of this video, I will share the key also. So let's see the first question. The number of protein subunits in CF1 component of chloroplast ATP synthase is. The options are 3, 5, 7, 9. The correct answer is 9. 9 subunits are present in CF1 component of chloroplast ATP synthase. Let's see the next question. Identify the following animals that have partially divided ventricle in heart. Partially divided ventricle in the heart. Okay. So the options are given: amphibians, turtles, snakes, crocodiles. So these are the 4 statements. Among them you have to pick the correct statements, any 2 of the correct statements. Okay, so options are 1 and 2, 2 and 3, 3 and 4, 1 and 4. So let's see the correct option. So the correct answer for this is 2 and 3. Option is the correct one. That means turtles and snakes are having partially divided ventricle in the heart. Let's see the next question. Match the following related to the nature of variability and the corresponding heritability in narrow sense. Okay, so here the 2 lists are given. We have to match them according to the correct sequence. List 1 consists phenotypic variability is entirely of genetic in nature. Phenotypic variability is entirely of environmental in nature. Half of phenotypic variability is genetic and half is environmental. Environmental component is 3 times the genetic component. List 2 consists 0, 0.25, 0.5 and 1. So here you have to match the list 1 with list 2. So options are given 1, 2, 3, 4, 2, 4, 1, 3, 3, 1, 2, 4, 4, 1, 3, 2. Okay, so I am going to match the list 1 with list 2. So observe carefully. Okay, so phenotypic variability is entirely of genetic in nature is related to the option 4, that is 1. Phenotypic variability is entirely of environmental in nature is related to 0. Half of phenotypic variability is genetic and half is environmental is related to 0.5. Environmental component is 3 times the genetic component is related to 0.25. So the correct option is last one, D, that is the correct one. Let's see the next question. Which is not a feature in penguins that will not fit into Darwin's concept? Options are huddling in large groups, monogamy, parental care by female, preening. So you have to pick the correct option. I'm going to give you the answer. Parental care by female is the correct answer. Let's see the next question. Here also you have to match the list 1 with list 2. List 1 consists plantation types and list 2 consists the category which are belonging to them. Okay, phanerophytes, cryptophytes, therophytes, chamaphytes. Okay, here you have to match the list 1 with list 2. So I am going to match the list 1 with list 2. Phanerophytes. Phanerophytes are related to plants where growing buds are located up the upright shoot much above the ground surface. Okay, so that is the correct answer. Cryptophytes, they are related to third one. The cryptophytes are the plants having bulbs or rhizomatous stem lies underneath the soil. They are called cryptophytes. Pterophytes, they are related to annual herbs which only survive by seeds. Chamephytes, they are related to first one, that is chamephytes are nothing but herbs or creepers whose buds are located close to the ground surface. They are called chamephytes. Okay, so let's see the next question. So here the correct answer is C is the correct answer. Okay, so let's see the next question. The scientist who first used the term survival of the fittest was. Options are Jean Baptiste Lamarck, Thomas Henry Huxley, Charles Robert Darwin, Herbert Spencer. So the correct answer is, so here we think the correct answer is Charles Robert Darwin. So that is exactly wrong answer. For this question, the correct answer is Herbert Spencer is the correct answer. Okay, so let's see the seventh one. So here also we are going to match the list 1 with list 2. This question is related to molecular biology. We have to match them accordingly. So Cleno, list 1 consists Cleno, T4 DNA polymerase, T3 RNA polymerase, RNase H. So I am going to match the list 1 with list 2. So Cleno is related to DNA polymerase that lacks 5' to 3' exonuclease activity. Cleno is related to DNA polymerase that lacks 5' to 3' exonuclease activity. So next one, T4 DNA polymerase. So that is related to that lacks 5' to 3' exonuclease activity. That lacks 5' to 3' exonuclease activity. Next one, T3 RNA polymerase is related to DNA dependent RNA polymerase. DNA dependent RNA polymerase. Last one, that is RNase H, which is related to used in the second strand cDNA synthesis. Used in the second strand of cDNA synthesis. So for this, the correct option is 4213 is the correct answer. So let's see the next question. Arrange the following steps in appropriate order to determine the presence of a specific allele in a DNA sample. So here also I'm going to give you 3 statements. Among them you have to put them in the correct sequence. Okay, so let's see the statements. PCR amplification of DNA. These are the steps, you have to arrange them in a sequence. Okay, PCR amplification of DNA, hybridize with allele specific probe, label the probe DNA. So these are the 3 steps, you have to arrange them in order. Okay, so I'm going to give you the answer for this question. B is the correct answer. That means PCR amplification of DNA, that is the first step. Next one is label the probe DNA, that is the second step. And third step is hybridize with allele specific probe, that is the third step. So next question. In a DNA chain, the nucleotides lying adjacent to each other are rotated by dash relative to each other. Therefore, the DNA double helix completes one full turn nearly by every dash base pairs. Okay, so here I'm going to give you the answer, that is so D is the correct answer. Okay, so let's see the next question. The number of transmembrane protein subunits, they are called connexins, that composes each connexon of a gap junction is. Options are 6, 5, 4, 3. So the correct answer for this question is 6 is the correct answer. Let's see the next question. Here also you have to match the list 1 with list 2. Here the types of mutations, genetic code, introns, alternative splicing, they are given. So frame shift mutation, degeneracy of genetic code, intron, alternative splicing. You have to match the list 1. These are the present in list 1. You have to match them to the list 2. So I'm going to give you the matching. Frame shift mutation. So in the options, the related option for this is addition or deletion of base pairs in numbers other than 3 into the coding sequence is a correct option. So degeneracy of the genetic code, which is related to single amino acid specified by more than one codon, that is the characteristic of the genetic code. That is called degeneracy of the genetic code. Third one is intron, which is related to the second option, that is a sequence of base pairs in a gene that is not represented by any bases in the mature mRNA. Okay, so the fourth one, that is alternative splicing, that is related to produce different mature mRNAs from the same primary transcript. Okay, so here the correct option for this question is first one, 4, 1, 2, 3. Okay, so let's see the next question. Pathogenesis related proteins are crucial in mediating plant pathogen interactions. List 1 indicates the list of PR proteins, while list 2 signifies their corresponding functions. Match the following. So, okay, pathogenesis related proteins, nothing but PR1, PR2, PR3, like that. We have to match them with the list 2. Okay, that signifies the functions of corresponding PR proteins. Okay, so I'm here the given proteins are PR2, PR4, PR5, PR12. Here we have to match them according to their function. Okay, so I'm going to match. So PR2 is related to beta 1,3 glucanase function. PR4 is related to chitinase 1 and 2. PR5 is related to chromatin-like protein. PR12 is related to lipid transfer protein. So here the correct answer is the first one, that is 2, 1, 4, 3 is the correct answer. So let's see the next question. Syncytial mode of cell specification is one of the cell fate determination mechanisms predominantly present in. So syncytial mode of cell specification is one of the cell fate. So let's see the next question. Syncytial mode of cell specification is one of the cell fate determination mechanisms predominantly present in. Options are insects, tunicates, amphibians, mammals. So the correct answer is insects is the correct answer. So let's see the next question. Arrange the following events of photorespiration in correct sequence. So oxidation of glycolate, decarboxylation of glycine, oxidation of ribulose 1,5-bisphosphate, emanation of glyoxylate. So we have to arrange them in correct sequence. So 4 options are given. Here the correct answer is B is the correct one. That means first one is oxidation of ribulose 1,5-bisphosphate, and then oxidation of glycolate, and then emanation of glyoxylate, and then decarboxylation of glycine. So that is the correct sequence. Let's see the next question. Match the following. Here also you have to match the list 1 with list 2. Tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume. So we have to match these with list 2. So here I am going to give you the correct answer. So D is the correct answer. That means tidal volume is related to second one, that is 500 ml. Inspiratory volume, reserve volume is related to fourth one, that is 2000 to 3000 ml. Expiratory reserve volume is related to first one, 1000 to 1500 ml. Residual volume is related to 1200 to 1400 ml. So let's see the next question. A strain of Neurospora with genotype A plus is crossed with another strain of genotype Y plus. On appropriate analysis, it was found that 90 asci were of type A plus, A plus, plus plus, plus Y, while 10 asci were of type A plus, AY, plus plus, plus Y. The map distance between the two genes is. So the options are, first one is 0.01 map unit, second option is 0.05 map units, 0.1 map units, 0.9 map units. So here I'm going to reveal the answer. For this, the correct option is 0.05 map units is the correct answer. Okay, so let's see the next question. Match the list 1 with list 2. Here also the two lists are given. Australopithecus, first vertebrates, Paranthropus, genus Homo. So these are the components present in list 1. We have to match them with list 2. For this question, so the correct answer is D. Okay, so that means Australopithecus is related to Africa, and that means that is present in Africa. First vertebrates are reported during 500 million years ago. Paranthropus is present in East Africa. Genus Homo is reported 2.5 million years ago. Okay. So let's see the next question. In pollination of fig by wasp, the fig offers the wasp the following: seeds as food for the developing wasp's larvae, egg laying site, warm temperature for the incubation of eggs, protection. So these are the 4 statements. Among them we have to pick the correct statements. So for this, the correct option is C. That means 1 and 2 options are correct. That means, so seeds as food for the developing wasp's larvae, egg laying site. So they provide egg laying site for the wasps. Okay, so these are the correct statements. Okay, let's see the next one. Which of the following gives direction to the evolutionary process? Options are gene mutations, changes in chromosomal structure, changes in chromosomal number, reproductive isolation. For this, the correct answer is reproductive isolation is the correct one. Let's see the next question. Corn is immersed in the boiling water. It is then cooled. The solution becomes sweet. It is due to. Options are enzymes are inactivated in boiling water. Disaccharides are converted to monosaccharides. Monosaccharides are converted to disaccharides. Monosaccharides are converted to polysaccharides. For this, the correct answer is B. That is, disaccharides are converted to monosaccharides. So that's why the solution becomes sweet. Okay. So let's see the next question. Here also you have to match the list 1 with list 2. Chromosome walking, quantic, positional cloning, physical map. So these are the list 1 components. We have to match them with the list 2 components. So I'm going to give you the answer for this first. So that is A is the correct answer. So let's see the matching. Chromosome walking is related to fourth one, that is using a DNA probe from the end of one clone to isolate a clone containing adjacent chromosomal DNA segment. So next one is quantic, which is related to set of two or more partially overlapping cloned DNA fragments. Third one is positional cloning, that is related to second one, method of cloning gene without prior knowledge of its function or its protein product. So physical map, so that is related to a map showing the order of cloned bits of DNA. So this is the correct order. So let's see the next one. Negative membrane potential in mitochondrion is observed in the dash. Options are matrix, inner membrane, outer membrane, inner membrane space. So for this, correct answer is matrix is the correct answer. Let's see the next question. Transport of one solute depends on simultaneous or sequential transfer of a second solute in the same direction across a plasma membrane is. So for this, the correct answer is symport. Select the following the correct definitions of the terms induction and attenuation in the context of gene regulation. So here the four statements are given, so we have to pick the correct statements. Induction is an irreversible conformational change of a protein molecule. Induction is stimulation of protein synthesis by a specific molecule. Attenuation is negative regulation of gene expression. Attenuation is gene regulation involving premature termination of transcription. So among them we have to pick the two correct statements. So I'm going to give you the answer D. That means second statement and fourth statements are correct according to induction and attenuation. Okay, so let's see the next question. So neutrophil NADPH oxidase system defects are responsible for the production of. Options are Hashimoto's disease, leukocyte adhesion deficiency, celiac Higashi disease, chronic granulomatous disease. For this, the correct answer is chronic granulomatous disease is the correct answer. That means fourth one is the correct answer. Okay.